everyone, I'm Mike Ward and welcome to Conversations in Healthcare, a video series brought to you by DRG, part of Clarivate. This episode is one of a number that we are recording alongside On Helix, a digital conference that's been hosted by One Nucleus. In line with this, I'm delighted to be joined by Nick McCooke, a serial entrepreneur who has been at the forefront of developing one of the key technologies, next generation sequencing, that has catalyzed the biopharma revolution in the past 2 decades. So Nick is currently the CEO of Longas Technologies, a Sydney-based startup that is developing approaches to improve dramatically the performance of industry standard NGS platforms. So having been at the birth of NGS as a founder of Solexa, a Cambridge-based company that he floated on NASDAQ, and the company was subsequently acquired by Illumina. He was also Chief Business Officer at DNE, which has moved NGS into the hands of the clinician. So Nick, I hope you and those you care about are keeping safe and well, and thanks so much for joining me.
Mike, thanks very much. It's a pleasure to take part in this, and also thanks for the introduction.
Great. So, as I mentioned in that introduction, you were one of the pioneers of what we now call, next generation sequencing. I think it might be helpful for the audience if you could just explain what was the status of sequencing technology at the time, and what triggered the creation of Solexa.
Yes, it's an interesting question. We're going 20 years back, of course, a little while ago. We started setting up Solexa in 2000. And at that time, the Human Genome Project was very much coming to its final stages, and it was accelerating as well. It was during that later period when actually a lot of the sequencing got done. And that was because the latest version of the ABI sequencer became available. The machine originally developed by Hood, Smith, and Hunkerpiller. And that was a 96 lane capillary machine. And that's a machine that went into all the big sequencing factories like the Sanger, that were really churning away at the Human Genome Project. But I think a lot of people thought that was the pinnacle of sequencing in a way. But at that time, there were a number of small groups around the world that were thinking about how to take sequencing on to the next level. And these groups came at the idea, independently. I don't think they knew of each other's existence. But the general or the common theme to those ideas was massive parallelization. In other words, not trying really to improve each step, the efficiency from one step to another, but actually to do millions of interrogations in parallel. And that was really the whole secret of NGS, to massively parallelize the sequencing process. So I think the final ABI machine could do about 1 million bases a day, something like that. So huge improvement of what had gone before. But next generation sequencing took that to a completely different level. So the team, founding team at Cambridge, had the idea that you could create a single molecule array of DNA templates, a massively parallel array, and interrogate all of those with a chemistry. That's how Solexa came into being, really, to develop that technology. It needed a completely novel sequencing chemistry, which was invented and developed in the company. But as time went by, the idea of single molecule arrays, we really struggled with that. We hit a brick wall, in fact, we couldn't take it any further. And that's when another group came into play. We found a technology developed by a group in Switzerland, and we acquired that technology. And really the platform that got developed and launched was a combination of what's called the Solexa chemistry, the sequencing chemistry, and the array technology that we acquired from the Swiss company.
Right, and when was that acquisition?
That was probably about 2004, I think. Yeah, yeah.
Okay.
So it's thought of as being Cambridge technology, but the truth of it is it's a Cambridge and Swiss technology. And I tell that story really to some degree to even up the landscape in terms of giving credit to all the people that were involved in developing the platform.
Oh yeah, so putting some flesh around the Solexa myth that was created. So you took Solexa though, you floated on NASDAQ, and then you left the business, and Solexa was acquired by Illumina. Over that period of time, from the creation to flotation, with that obviously that subsequent acquisition, what had Solexa achieved? What was it that first allowed you to get that NASDAQ flotation, and then obviously what attracted Lumina?
Yeah. So at the time of it was strictly a NASDAQ listing because we reversed into a listed NASDAQ company rather than floated, but the same effect. At that point, we'd acquired the Swiss technology, and actually, once we acquired that, we started to make very rapid progress applying our chemistry to their array technology, and we'd sequenced Phi-X, which is the rites of passage organism that all companies developing sequencing technology sequence, which is about 5,000 bases, something like that. So we'd done that. So the technology was working. But it was very much at a breadboard stage. So that's where we were when we listed, and then the company effectively became an American company listed on NASDAQ and with an operation in California. And the next stage was development of the instrumentation. And by the time of the Illumina acquisition, the first product had been launched. I mean, very much a beta instrument, but it had been launched. There was an instrument out there with a Solexa label on it.
Yeah.
So Lumina were able to see a technology which was essentially working, lots of bugs and all the rest of it, but basically a working technology.
Yeah. So yeah. I mean, you weren't there at the time of the Lumen acquisition, but it'd be interesting to get your thoughts on, what was it that, the acquisition of those technologies that were, housed at Solexa. What did that allow Illumina to do with NGS?
Yeah, well, it completely transformed Illumina. I mean, they were an array company, actually doing quite well, but their business became a sequencing business. So what they were before continues even to this day, but of course their business is completely driven by the sequencing business. So it transformed them as an organisation completely.
Yeah. But in terms of the application of NGS, because with the Human Genome Program, sequencing there, it was very much a research tool, and that was, certainly it appeared in the early days of NGS. It was a research tool. Were they able to do, push the markets into a different place?
Oh, I think they did a number of things. I mean, yes. I mean, it started as a research tool. I think it is still a very important research tool as well. I think maybe clinical applications grab the headlines, but I don't know the numbers. But I would imagine probably sales into the research environment probably still dominate. But the future clearly is a push into the clinical applications, and some of those have been around for a few years now. So I think Lumen did two things. Two big things. I mean, they continued to improve on the technology. So what it's capable of doing today is, far more than was at the time of the acquisition, and they've broadened the range of applications. And yes, particularly as you point out, moved into the clinical space, which is very important. And I suspect their value today is partly attributed to the opportunity they have going forward into the diagnostic space.
Sure, and I mean, so thinking about, so when you were there at this sort of the foundation of Solexa, you would have got your business plans, potential addressable market. Did it develop the way that you were anticipating, and for example, with what Illumina's been able to do?
I think the honest answer is we didn't see it in its entirety, and I don't think anyone did. And to be honest, I think if anyone says they saw it all from the beginning with great clarity, I simply wouldn't believe them. I don't think anyone did. We knew we were working on something important, something big. We had that sense, but could we see all the varied applications that it might be applied to? No, I don't think we did, and I don't think anyone did. We knew it would be transformative, but we didn't know the specifics of how. Yeah, sorry. Yeah, go on. I was going to say, company meetings, we talked about it. We talked about working on something of great import, but so we had that sense, and the privilege of working on something like that. We definitely had that, but we couldn't see all the details. I mean, essentially, it was a revolutionary tool which, put into the hands of creative scientists all around the world, led to explosion of new projects and applications and so forth.
Yeah. So you mentioned about sort of clinical use. These days, a lot of life sciences companies talk about being patient-centric, putting the patient at heart. I just wonder, next generation sequencing, has it sort of provided any sort of tangible benefits to patients that actually patients might be aware of?
Well, there's nothing more personal than the genome, so it is at the heart of personalized medicine, and I think a lot is hidden because a lot of its use goes into the development of into drug development, and people may not see the connection between the drugs they're given and NGS. But as a diagnostic tool, of course, it is coming much more to the fore. So, a cancer patient, it could be involved in diagnosis, prognosis, choice of treatment, and follow-up post-treatment. So NGS could be involved in the whole patient journey there. So I think it is something that people are becoming, well, if they don't necessarily know that the technique is used for diagnostic or so forth, maybe they're not familiar, but they're certainly becoming engaged with the technology, whether they know it or not.
So in 2020, we're seeing a lot of the life sciences industry, redeploying, its resources, its assets to tackle the sort of challenges unleashed by the sort of COVID-19 pandemic. What role might next generation sequencing have in the sort of development of solutions to the pandemic?
Well, it's been involved from the beginning, really. The sequencing of the the viral genome, very, very quickly, of course, was done using NGS. Tracking the mutation of the viral genome as it spreads around the world is being done with NGS. The diagnostics, derived from the knowledge of the sequence, so that's relevant to that. I'm sure there's drug development going on, which is, which uses knowledge derived from sequencing. So I think it's just, it's typical. NGS has become just part of the fabric of the life sciences now. It's an essential tool, and so any endeavor really in the life sciences is likely to use NGS at some point. It's a little bit like PCR, which became ubiquitous. You can't imagine doing work in the biological sciences without using PCR, and I think the same is true of NGS now. It's just a ubiquitous, universal tool.
Yeah, and of course, as I said, you were there at the start, so you've seen the evolution from it, going from research into sort of more these clinical, these diagnostic applications. You're now CEO of Longas Technologies. What are the sort of the technical challenges that Longas is seeking to address, and what do you think the solution is to those challenges?
Yes, so the world of sequencing, well, you can divide it in lots of different ways, but one of the ways people divide it commonly is between long read sequences, sequencing platforms that can read long lengths of DNA, and short read sequences. And the dominant technology in the market is the Illumina sequencing platform, which is a short read sequencer. Back in Solexa days when we developed the technology, that's just an inherent part of that way of sequencing. You sequence 150 bases, something like that, fragments of 150 bases. Now, I think for lots of applications that's absolutely fine. You don't need more than that, and it's extremely accurate and it's very cost effective. So it's a very attractive platform, and that's why it's been very successful. But there are occasions when you do need to generate long reads, and the Illumina platform can't do that. So when you want to look at structural variation, you want to distinguish homologous regions of the genome, you want to get into repetitive regions, etc. There are lots of applications where long read is really what you want. So our thought was, well, how would it look if we could basically allow all those short read sequences out there to become long read sequences when you needed to do a long read project or experiment? So that's what we've done. We've developed a technology which effectively converts short read sequences like Illumina, but others too, to behave as a long read sequencer, so they can do all those things I talked about a moment ago.
Right. So and how long is a long read?
We've been generating reads using an Illumina sequencer as an example of about 10 kilobase. So that's 10 kilobase compared to 150 bases. So it's a huge difference. Once you get over about 7,000 bases, something like that, 7,000 to 8,000, you can really then start to get into the dark matter, if you like, of the human genome. There's a sort of critical length there. So we're well over that.
So dark matter of the human genome. Do you want to sort of define what you mean by that?
Well, if you look at it from the perspective of an Illumina sequencer, it basically means those parts of the genome you can't resolve using short reads. So they are the repetitive regions. They're the regions with, homologous sequences, where you get structural variation. To be able to locate that, where it actually should be placed, requires long reads. So those are the sorts of pieces of work which benefit from having a long read capability. So with our technology, someone who's sitting there with an Illumina sequencer can actually just prepare it and operate it in a way which allows the scientist to generate long reads for that particular experiment.
Right. So I mean, you can literally sort of take your technology and almost plug and play it with the sort of the kit that they've already got on their lab bench.
It's a different kit, and it requires different data processing. But other than that, you can use the sequencer you already have sitting on your bench. You don't have to go out and buy a long read sequencer or send your sample away to someone who can run a long read sequencer. You can do it all yourself. So
okay.
Yeah.
So and where are you in in the sort of development of doing these long reads? I mean, is this something that is now already sort of fit for purpose, or do you still have?
Yeah, it is.
Concepts?
I mean, it's not on the market yet because we've decided we want to go to market via a partner. So we're in the process of seeking the right partner. But in terms of working, yes. I mean, we've demonstrated that we can generate sequences of whole genome sequences from bacteria at finished quality, which is the ultimate test, if you like. And we've sequenced human genomes with average read length of 10 kilobases, and we've shown that we can identify variations, for example, that you wouldn't be able to determine with short reads alone. So in terms of working, it works. So there really isn't more to do to demonstrate that. It can be improved, and of course we're carrying on improving it, but it works extremely well. The nice thing actually is that the underlying quality or accuracy of sequence using Illumina sequencing chemistry, the original Solexa chemistry, is extremely good. So using that in a long read modality gives you a really nice result, the best of both worlds, in fact.
Right. And I mentioned it that the company's Sydney based, right? So
Yes, yeah.
so what's the what is the sort of the anticipated business trajectory for Longas?
Yes, people usually assume I'm based in Sydney when they see I'm the CEO of the company, but so I was working in virtual mode long before lockdown. The goal is to partner. We're looking for partners who are able to, who have international reach and are able to take the product effectively to market. We think that's a better approach than trying to build the business out from where we are at the moment. Right,
but the technology was originally developed in Australia.
Oh yes, it was. It was developed in a Sydney-based University of Technology, Sydney. So that's the reason it's there. Yeah.
Okay. So as we look to the future, and you get the crystal ball out, what do you think next generation sequencing might actually be able to deliver that we haven't got to at the moment? And what would actually need to happen to be able to fulfill that kind of dream?
Yeah. Well, you're asking someone who wasn't entirely accurate back in 2005 and 6, but I'll have another go. But actually, in 2002, I was interviewed by the BBC, and I predicted we'd reach $1,000 genome. That's bettered now. Actually, I got into a lot of trouble at a conference for predicting that. They thought I was being a bit too wild. But I also said in that interview, I thought the time would come when the human genome our own human genome, our personal genomes, would be part of our approach to personalized healthcare. Worse that effect, anyway. And I still think we've got further to go. I mean, it's not as simple, of course, as the way I couched it at the time. And it's perhaps not as much a technology challenge as one of interpretation and how we use the information. But it seems to me we haven't got to that point that I predicted, and I personally, would like to see, that this thing we have, our own personal genome, being used more and more in the way we manage our health, or other people help us manage our health. And I think we have way to go there yet. So.
Yeah, because of course the original cost of the Human Genome Program was a phenomenal amount of money. So 1,000 $1,000 genome in 2002 might have seemed to be, I can imagine being, wow, that's a
I
small
think
cost.
people thought that
But,
was bad. Yeah.
but how much does it cost now?
I think it's in the hundreds. I'm not familiar, but I think, you know, if you use the right machine in the right way, you're down to, you know, 100 or so.
So you're definitely in the right order of magnitude. So,
Yeah.
so that's good. So one final question. I in the introduction I mentioned that you were a serial entrepreneur. You've been there at the start of companies. Knowing what you now know, if you were able to go back in time, what advice would you give to the young Nick back in 2000, in 1999, when you first sort of came across this approach?
Yeah. Well, I mean, in the sense of, you know, regrets or not, I mean, a general philosophy of life, I don't have regrets for anything I've done or didn't do. I mean, I think that's very important. I don't see the point. But pressed for an answer, it would probably be, mate, I'd hang on to you Illumina shares for just a little bit longer than you did.
Yeah. Well, yes. I mean, that's hindsight is always 20/20, as they say.
Yeah.
But
I don't have any regrets.
but I was just sort of thinking, in terms of, for young entrepreneurs today, was there something that you, say, for example, did back in 2000 that actually you think, wow, that really helped us out there, that I would do that again and again and again?
Yeah, I think there are two things. I mean, surround yourself with the best. I mean, these are a bit cliched, of course, but surround yourself with the best people you possibly can. I mean, that's just got to be, you know, the best thing you can do. And the second is, you know, be really stick at it, you know, because there will be many bad moments on the journey. Many good ones, but there'll be many really tough ones where you're really challenged to, you know, your faith in the project is challenged, and you really have to be strong and seek, you know, and get through those points, because if you waver the first time you get to one of those crises, then it's all over.
So as a serial entrepreneur, does it get any easier, or do you still always have the sort of the self-doubt of, is this what I should be doing?
Well, I think so. You know, I think a bit of self-doubt's healthy. I mean, it's a mixture of, you know, challenging, but also, you know, determination and getting that mix right. But no, things don't get easier, because I think if things look really easy, you've almost certainly missed something.
Yeah.
You know, it's the ease and confidence of ignorance, I suspect.
So great, Nick. Yeah, as always, it's been a great pleasure talking to you today. Yeah, I think the insights that you provided around NGS will be really interesting to people, because, you know, these days, you know, people kind of take it for granted, and yet, as you say, 20 years ago, it's something that we didn't have. So thanks very much for taking the time. And, you know, if you'd like to tune into future conversations, follow our LinkedIn page, because that's where we will be posting alerts to future releases. So in closing, I'd like to thank Nick again for joining us, and thank everyone for tuning in. It's great to have these audiences. So until next time, stay safe and healthy. And I'm Mike Ward, and I'll see you in the next episode.